Good morning, all of you. Welcome to our session. Today, we'll be discussing question from Biotechnology 1, Principle and Procedure. Okay? The most probable questions from this chapter we will be discussing today. Okay? Biotechnology is given more and more importance in Neet examinations right from 2016 onwards. We get more questions, more proportion of questions from the biotechnology 2 chapters in these years. So you give that much of importance to these chapters. Okay? Right away, I am going to the questions. Question number 1 on your screen. Genetic engineering. What is genetic engineering? Engineering with the genes is essentially dealing with what are the important features of genetic engineering, technique to alter the chemistry of genetic material. We are engineering with the genes. What is engineering? Bring deliberate changes according to our will. You manipulate in such a way that it brings your desired changes. Desired changes are brought about to what? To the genes. That is why it is called genetic engineering. So, the first option. 2. Maintenance of sterile ambience. See, sterile ambience is a part of chemical engineering, whereby we are making cultural medium which will not be damaged because of contamination. So maintenance of sterile ambience in chemical engineering process to enable growth of only the desired microbe or eukaryotic cell in large quantities. It is only a tool that enables the growth of genetic engineering. Genetic engineering grew mainly because of the contributions of this part of chemical engineering. Creation of recombinant DNA, that is the most important process. The most important event in genetic engineering is rDNA technology. We are all taking desired DNA that is responsible for our desired quality, desired character from one organism which is called the donor, and give that gene to another one to which you want to have this character given. That is the most important technique in genetic engineering. So creation of that recombinant DNA, use of gene cloning and gene transfer. So both A and C are the correct options. Restriction enzymes. See, a genetic engineer need a number of tools. The most important tools a genetic engineer should have are 1. Enzymes, okay, then vectors, etc. Then one of the most important enzyme is restriction enzymes. Why they are called so? Because they are all restricting the growth of foreign DNA that enter into the bacterium. What is that foreign DNA? DNA of the viruses. Remember, viruses are abundant, bacteria is also abundant. Viruses are attacking bacteria and are destroying the bacteria. Then that is the case, there should be no bacteria living now. All bacteria should have been removed by the attack of viruses, but it is not happening. Why? Because viruses are not allowed to multiply within the bacterium always. Bacterium is having an ability to resist, to restrict, to limit the growth of their DNA within the host cell, within the bacterium cell. That's why the bacteria are also now living. Okay. Werner Arber thought like that. There must be a mechanism present within the bacteria. That's why they are all still living. they are capable of restricting the growth of viruses within them. And this principle they discovered in the year 1963. They called it as restriction endonuclease because it is an endonuclease that is restricting the growth of the viral DNA within a bacterium. So they have the ability to limit the number of strains of bacteriophages infecting the bacteria. Clear? That enzyme is having an ability to restrict the growth, multiplication of the viral DNA within a bacterium. That is why they are called restriction endonuclease. Actually, these restriction endonucleases are a part of an enzyme complex. I will be telling you. The two enzymes that form that enzyme complex are one, methylating enzymes that will add on methyl group to the nitrogen bases when the DNA is synthesized. That's called methylating. They go on adding methyl group, methylating. While the other one is cutting unmethylated DNA. That's called restriction. These two enzymes are forming a complex called methylating restricting enzyme complex. That methylating restricting enzyme complex is responsible for adding methyl group to the DNA nitrogen bases so that nitrogen base is not attacked by its own restriction enzymes because it is methylated, it is not available for cutting. Thus, its own DNA are all protected. Whereas unmethylated, who is unmethylated? All the DNA that are produced within the host cell, within the bacterium cell will be methylated. So, which will be unmethylated? The DNA that is coming from outside, that is the viral DNA. Since it is not synthesized inside the bacterium, it is non-methylated. That non-methylated will be identified and cut by the second enzyme. That is why it is called restriction endonucleases. So, it is forming an enzyme complex consisting of a methylating, second one restricting. That is why this complex is called methylating restricting enzyme complexes. Okay. Add methyl group to the host DNA, its own DNA, so that its own DNA is never cut, it is never damaged, but it will be cutting bacteriophage DNA, the DNA that is coming out, coming from outside. That is, they are not at all methylated, hence they will be cut. Elution. What is the elution principle? Elution is the process whereby from the agarose gel, suppose this is the restricted enzyme fragment which contain our desired DNA, we cut it with a sterile instrument, take that chip, small piece of that agarose gel and put it in a suitable solvent so that gradually the DNA get dissolved and thus is transferred into that solvent. Okay, this process of isolating and separating from the chip, from the agarose gel to the medium is called elution. So bands of DNA are cut out from the agarose gel and extracted from the gel piece. We are cutting this gel piece, putting it in a suitable solvent so that it is converted into this solution. This is called elution. In order to link an alien DNA is foreign DNA from outside, the vector need to have very few, preferably single recognition site. Why we need only very few, preferably one? Suppose you are having a number of cutting sites, recognition sites for the particular enzyme. The same enzyme is having more than one cutting site. Then what will happen? It will cut it in all these pieces or cut it the DNA into so many small pieces so that some piece may be lost, some piece may be introduced in a inverted order, etc. So the entire process is likely to be damaged. There is more likely that we may have some mistakes, okay, because the vector is cut into so many fragments. We don't want to complicate the process. If it is cut into so many pieces, the entire process will be complicated. So to avoid that, we need only one restriction cutting site for a particular enzyme in the vector. Okay. So vector with multiple cutting sites, that will be complicating the entire process. Alternative selectable markers with chromogenic substrate. This is what is given in our book. Here, in this method, we are using new type of plasmid, new generation of plasmids called PUC, discovered by University of California. It's a plasmid, artificially prepared hybrid vector, and this will be having its own ori and a lakhs gene. Close to the lakhs gene, you will be having multiple cloning site, meaning cutting site for a large number of enzymes. What are the cutting site of enzymes, or what enzymes can cut it? That is determined, and so there are so many different types of PUCs. These are all in pairs, and there is a selectable marker, namely ampicillin resistant gene. And regarding that, which differentiate recombinant from recombinant, select the true statement regarding the selection procedure. Suppose you are using this one, you have to culture in a medium, only in one. That is the advantage. If you are using PBR 322, we have to culture in two media simultaneously. Okay, plating in two different media simultaneously is a cumbersome process, difficult process. That's why we have developed new technique, and this is having some advantage. We need culture them or plate them only in one cultural medium that contains ampicillin and a coloring agent which is called X-gall. X-gall is a chromogenic substance similar to that of lactose. Okay. Here you are having lakhs gene. Lakhs gene we are studying in lac operon. That gene is taken, it is of E. coli itself, and we are having beta galactosidase enzyme produced by the lakhs gene, which will be acting on this substance, chromogenic substance called X-gall, and the X-gall is converted into colored products by that enzyme. Okay, beta galactosidase. So, if this gene is active, the X-gall will be converted into colored products. So, the colony will be growing as colored, which indicates that this gene is active. When this gene is active, it is not cut by a cutting site or cutting enzyme, and no foreign DNA is taken. It is a non-recombinant transformant. Okay, we want a transformant recombinant. That means which is cut here, anywhere a foreign DNA is introduced. Okay, and because of that introduction of the foreign DNA, what happened? The lakhs gene is insertionally inactivated. Since lakhs gene is insertionally inactivated, it is not working, it is not producing the enzyme. X-gall is not at all converted into colored product. So the colony will be growing without color. That is what we want. It is the recombinant transformant. That's how we are all selecting. Okay, that is the selecting procedure, screening procedure. So you go through. A non-recombinant colony have the ability to produce color. Non-recombinant, because now foreign DNA is introduced, the lakhs gene is working, so it is producing the enzyme, converting the X-gall in the medium into colored products. The recombinant DNA is inserted within the coding sequence of an enzyme, okay, beta galactosidase, and results in the insertional inactivation. In recombinant DNA, foreign DNA is introduced because multiple cloning sites is within that lakhs gene, and colonies that do not produce any color are identified as the recombinant colonies. That is what we want. They will be growing without any color. Okay, that's also correct. So all of them are true. Place in order the following steps involved in PCR. See, what is PCR? Polymer chain reaction. In polymer chain reaction, we are all having a thermocycler machine. Remember, PCR is a technique that enables us to produce large number of desired DNA or gene in vitro conditions. We need not depend on any organism for amplifying our desired gene. It is having only 3 basic steps. What are these basic steps? In the first step, I will be giving you diagrams later on in subsequent questions, but just for understanding. Initially, the temperature is raised to 94 degrees. When the 2 strands of our DNA that is to be amplified, that is called template DNA. The template DNA is denatured. It is melted. The 2 strands are separating. Melting of the template DNA, denaturing. In the second step, step B, the temperature is brought to 50 to 60 degrees. Automatically the machine, it's a computerized machine, which will be automatically undergoing temperature changes. That is why it is called thermocycler. Now, in the low temperature, primer molecules are attached, primer annealing. And the third and the last stage, the temperature is raised to 72 degrees Celsius. At this temperature, Taq polymerase start functioning and polymerization starts chain elongation. Okay, so by that time, 1 become 2 chains, 2 become 4 chains, like that. It is amplified. Okay, so what are the procedures? Newly synthesized strains act as template. That is the next and the last stage. Okay. Temperature is lowered, DNA primers and polymerases are added. Heat separates the strands of target DNA. What is the target DNA? That is the template. So that should be the first one. Okay. Now, once the 2 strands are separated, the temperature is lowered and primer is annealed, DNA primers. So it should be the second one. Then complementary base pairing between primers and template. That is what is called primer annealing. And DNA nucleotides are added in this next stage, that is Taq polymerase, chain elongation, and finally newly synthesized strands act as templates for the next one, that is the 5th stage. Okay, so these are the 3 basic steps in PCR. Carey Mullis, the great American, discovered this technique, and because of that, now we are having a very easy and simple method of producing large number of any gene, any copies you can have. Okay, that's why now we are all capable of having any copy of any gene. The significance of heat shock method. What is that heat shock method? It's a technique in making the host cell to take a DNA from outside. That is called competency. We have to make the host cell be competent. A bacteria cell will not be taking. This is the host cell. We want to give a desired DNA to this one, but Our DNA cannot enter into this one because it is having a cell membrane. That membrane is impermeable to water soluble DNA. It is allowing only fat soluble materials to enter in. Plasma membrane is lipophilic. It will allow only fat soluble material to enter in, not water soluble DNA. So we have to make a technique. Okay, what is that? It is called competency. You treat it with bivalent cation first and keep it in low temperature. So first of all, we keep it in bivalent cation like calcium, then keep it in ice, and suddenly you keep it to or transfer it to a higher temperature of 42 degrees. Again back to ice. So repeatedly you change the temperature so that because of that quick sudden temperature changes, there may be some transient pores, temporary pores produced in the membrane through which DNA may enter in. That technique is called competency. Okay, this technique is called heat shock method. Binding of DNA on the cell wall, no. Uptake of DNA through the transient means temporary pores in the bacterial cell wall. Through that, the DNA will be entering in. Okay, that technique is called making them competent. If a recombinant DNA bearing gene for ampicillin resistance is transferred to E. coli cells, and these host cells are spread on agar plate containing ampicillin. See, our DNA, recombinant DNA is having gene for ampicillin resistance. They are having ampicillin resistance. And if they are all culturing in a medium containing ampicillin, no problem. The host cells are now getting the ampicillin resistance. They are having an ability to grow in an antibiotic containing medium, ampicillin containing medium. So they will grow. So only a transformant. What type of transformant? A recombinant transformant will grow. Okay. Whereas non-transformant will not grow. So a transformant recipient cell will grow and non-transformant will die. See. This is the bacterium cell. And this is the vector. And this is the desired DNA. Now, this is the vector and this is the desired DNA. This has received the recombinant DNA from outside. Now it is called a recombinant transformant. Why it is a transformant? Because it has taken the DNA from outside without an agent. Transformation is a technique or a process whereby a DNA is entering into a host cell without an agent. We have studied bacterial transformation, Griffith effect, etc. Yes, same thing. So DNA has entered into, and that DNA is a recombinant DNA. It is having the DNA of our interest plus an agent, that is a carrier molecule, that is a vector. Now this is a recombinant transformant. This recombinant transformant is having the ampicillin resistance. This is having ampicillin resistance. Okay. So that it can grow in a cultural medium that contains ampicillin. So it will be growing. Whereas those who have not taken that recombinant DNA will not have the ability. Because E. coli, which is generally the host in our biotechnology of engineering, genetic engineering researchers, E. coli is having no natural ability to resist any antibiotics. By nature, this is having no ability to resist any antibiotics. Clear? So that it cannot grow. Now, which of the following statement is not true? DNA being hydrophilic. What is hydrophilic? Water-loving molecule cannot pass through the cell membrane. True statement. Okay. Agrobacterium tumefacient deliver a piece of DNA known as zDNA. No, this is the Agrobacterium tumefacient. This is having TA plasmid, tumor-inducing plasmid. That tumor-inducing plasmid is having a small segment called tDNA, which is responsible for making the cells, plant cells, cancerous. Okay. Once it is introduced into the host cell, which is a dicot plant, only the plasmid will be entering in and it will transfer that tDNA into the nucleus and is integrated into the genome. It is made a part of the genome of the host. So Agrobacterium tumefacient deliver a piece of DNA known as tDNA, not zDNA, in the TA plasmid, which transform normal T cells into tumor cells. Okay, so that is wrong. Retroviruses and adenoviruses are also used as cloning vectors in animals because of their ability to transform normal cells into cancerous cells, because they can multiply. Okay, that's why they are all used. In genetic engineering, DNA from different sources are cut with the same restriction enzyme so that both DNA fragments have same kind of sticky ends. They will be having complementary sticky ends. That is the principle why we are all cutting the vector DNA and donor DNA with the same restriction enzyme so that they will be having complementary cut ends. They can be easily be joined. A bioreactor provides optimal condition for achieving the desired product, correct? That is why we are all providing all the conditions favorable, temperature, pH, substrates, salts, vitamins, oxygen, etc., everything correct, providing optimal growth conditions so that the whole cell will multiply in an exponential way. So without any break, they will be going on multiplying. We need not have to stop the reaction. It will go on. Continuously, materials are provided and continuously used cultural growth is removed from it. We are having facilities for adding cultural medium fresh and taking out used cultural medium from a sampling port. Clear? This is the two type of, actually we don't want this diagram now. A stir tank bioreactor have been designed. This is the stir type, simple type. There will be a stirrer which is continuously moved using a motor and it is having impeller, okay, flat bladed impeller which is slowly stirring the content so that the content is having a uniform consistency. Oxygen will be reaching every part. That is the main function. It is designed for purification of the product. No, purification will be done only after taking out. That is a part of the downstreaming process. Addition of preservatives, no. Ensuring anaerobic, it is aerobic. Availability of oxygen throughout the process. Uniformly, oxygen is made available to every corner, every part of this one. And easy mixing is facilitated by a curved bottom also. Okay, the design is like that. The bioreactor will be having a curved bottom to make the easy mixing of the content. For addition of foam breakers, no. So the most important is that oxygen is made available. That is why we are all continuously stirring so that homogeneous condition is made available to all parts, all cells. There will be billions and billions of cells and all of them are given same condition, optimal condition always. The sequencer controls the copy number. What is copy number? The number of copies of a plasmid that is living or that is present in a host, that is called its copy number. Sometimes it may be low copy number, sometimes it may be high copy number. Who controls that copy number? Copy number is controlled by a segment called ORI, origin of replication, from where replication starts. The same replication starting codon is also responsible for the number of times it should multiply. That is controlling the number of copies. Okay, so it is ORI site. What is a palindromic sequence? Palindromic sequence is a sequence that will be recognized by a restricted enzyme. And recognition sequences are all palindromic sequences. And selectable marker is a site which is used for selecting transformants from non-transformant. That enables the researcher to select which one is the transformant and which one is not a transformant. And actually this is a question from the next chapter which is not correct. Proinsulin has an extra peptide. See, proinsulin is having three parts. First part, the last part and the middle part. The proinsulin has an extra peptide called C-peptide, which is removed during maturation. That's the correct statement. The functional insulin has A and B chains linked together by hydrogen bonds. No, disulfide bond, SS bond, disulfide bond. It is not hydrogen bond. Genetically engineered insulin produced in E. coli. Yes, we will be studying it in the next chapter. It is prepared by producing alpha chains and beta chains separately and mixing them together. Genetic engineered insulin is produced in two types of E. coli cells. In man, insulin is synthesized as a proinsulin. What is a proinsulin? An inactive insulin, which is having the middle C-peptide. That is to be removed. Once it is removed, okay, it will be working. Alpha and beta will be joined together by disulfide bond and that will be functioning. Okay, so all of these are correct, but the functional insulin A and B chains are held together by disulfide bond, not by hydrogen bond. In gel electrophoresis, separated DNA fragments can be visualized. How can you see the DNA fragments? This is a glass plate on which you have made a thin film of agarose gel, and the medium is helping us, the medium in which the DNA is running, and depending upon the sizes, they are all running at different speed. The smallest one will be moving very fast, so they will be reaching the anode faster. We are loading in the well which is very close to the cathode. Why the molecules are running towards anode? Because nucleic acids are negatively charged. That's why they are all attracted towards the anode, positive charge. And how you can see them? We have to stain with a ethidium bromide, which is a fluorescent dye. Since it's a fluorescent dye, we have to observe under UV light, because ethidium bromide is a fluorescent dye. It can be seen only in UV light. That is why we have to observe it, watch it in UV radiation. Wrong statement with regard to restriction enzyme. They cut the strand of DNA at palindromic sequence. Each of them will be having its own recognition site, which is either 4 or 6 nitrogen base in size. And after recognizing a site, it will be cutting both strands. Okay. So they will be palindromic sequence. What is a palindromic sequence? Which will be read the same, in same direction in both strands. Remember, the two strands of DNA are anti-parallel. Okay. We have studied that GAA, TTC, GAA, TTC. If you are reading from 5 to 3 end, you have to read it, the complementary strand also in the same direction. So we get GAA, TTC, GAA, TTC. Now if you are reading from 3' end, CTTAAG, Then also you read in the same direction the complementary strand, again you get CTT-AAG. That is why it is called a palindromic sequence. They are all useful in genetic engineering. Restricted enzymes are one of the important tools because they are all acting as molecular scissors or scalpels. Sticky ends can be joined together by DNA ligase. Ligase is the joining enzyme, it is the molecular glue. Each restricted enzyme functions by inspecting the length of the DNA sequence. It is recognizing a particular sequence, whether they are having their own characteristic particular sequence, which is called its recognition sequence, and that is a palindromic sequence and will be cutting at a specific site on it. Okay, so regarding restricted enzymes, the last one is not correct. Again, the specific palindromic sequence which is recognized by EcoR1 that I have drawn earlier, it is GAATTC, GAATTC. Okay, and the cut is between G and A. The cut is between G and A, so that one chain will be small, the other chain will be long. That is called staggered end cutting, long chain cutting. And Herbert Boyer was an expert in this type of restriction enzyme. Okay. DNA molecule to which gene of interest is integrated for cloning is called. To which you should give that desired DNA. We need a carrier molecule. That carrier molecule that takes our desired DNA is called the vector or agent. Vector means an agent that is helping in transferring one DNA piece from one organism to another organism. Okay, we are taking the DNA, desired DNA from a donor and we want to give it to a host. But for that, we need the help of a agent, a carrier molecule. That carrier is called vector. Usually, the vectors are plasmids, viruses, okay, in early stages. Nowadays, we are using the even retroviruses if we want to transfer it to animal cells or agrobacterium tumefaciens vector, that is TIE plasmids, as an effective agent in transferring. We are also having a large number of artificial hybrid vectors produced, synthetic vectors, okay, recombinant vectors produced nowadays. The cutting of DNA at specific location became possible. Why? Why we could cut any DNA? Because we are having the efficient cutting scissor, scalpel, molecular scalpel or molecular scissor. What is that? It is the restriction enzyme. In our book, it is given from more than 230 sources. There are large number of more than 900 restricted enzymes are isolated and purified that we can use for joining, cutting and joining. Okay, so they are the molecular scissors or molecular scalpels, restriction enzymes. Ligases are molecular glue for joining DNA of different sources. And Prob is a single-stranded DNA or RNA, single-stranded, which is radioactively labeled to detect a counter molecule, a counter chain. Such a single-stranded RNA or DNA, it may be even antigen or antibody used for detecting a counter molecule because of that specific action is called a probe. Okay. And selectable markers are sequences which are all generally present in plasmids, which are all highly helpful in selecting transformant from non-transformant. We want to eliminate non-transformant and selectively clone culture transformant. That sequences or genes which are helping us for this activity is called selectable markers. Usually selectable markers are antibiotic resistance gene. Stirred tank bioreactor have been designed. We have seen that in earlier question. Availability of oxygen throughout the process. That is why we are having that system of stirring. Continuous stirring is required so that there will be oxygen available. All essentials are reaching every part of that bioreactor. Remember, a bioreactor will be having thousands of liters of cultural media processing simultaneously. So, it's a very huge vessel. All parts of that will be getting the same condition. So, the bacteria, the host cell can grow, they can multiply in an exponential way. They are all given all optimal favorable conditions. A foreign DNA and plasmid cut by the same restricted endonuclease can be joined to form a recombinant plasmid. For that, we are all joining. See, we are having a desired DNA taken from a donor. Okay, this is the donor DNA. And we are having a plasmid. What should be done? The mechanism is very simple. You take the same restriction enzyme, REN, same enzyme, cut it. Okay, so they will be cutting both of them with the same staggered end, same cut end, which are complementary. So after that, you mix it in a chamber. You put them so that these pieces are joining together. Okay, thus it will be forming. You have to add ligase. From where you are getting this ligase? Ligase is abundant in viruses. So you take that DNA sequence for the production of the ligase from a virus and give it to some organism so that it will be producing the enzyme. Get that enzyme. That is the molecular glue. So in some books it is given that ligases we are obtained from viruses. Then some children are having it out. So viruses are having no enzyme. They are having no machinery. Yes, that's absolutely correct. We take that DNA sequence which is responsible for the production of that enzyme and give it to some organism so that they will produce that enzyme for us. That enzyme is produced in some other organism using the machinery of some organism, but the gene is belonging to virus. Okay. Now, which of the following is not a component of downstream processing? What is downstream processing? From the cultural medium, we are all taking down the used cultural growth, the used cultural medium, from which we have to isolate, separate our desired protein. Recombinant protein is to be isolated, separated. Then it should be purified, and the entire process, starting from separation, removal of the used cultural growth from the downstream or taking out pot, downstreaming process, everything, all process, starting from taking out of the used cultural growth to marketing. That's called the downstreaming process. So we have to add preservatives to that, correct? Separation of the recombinant protein, that's a very important one. Purification, preservation, but expression. What is expression? A gene is expressing means the gene is producing a character. A gene is producing a protein. That is what is called expression. Already the gene has expressed. That is why you are having the recombinant protein produced in the cultural growth. That cultural growth is containing the recombinant protein that you are all taking down from the pot. You are all taking the cultural medium, used cultural medium. From that cultural medium that contains the expressed protein, that protein is produced because of the expression of the recombinant gene you have given to, and that protein produced is called recombinant protein, that is to be separated, purified and marketed. All of them are part of the downstream process, and expression is what is happening already, because of which the protein is produced in the cultural medium. Which of the following resident enzyme produce blunt end? See, all enzymes that are all mentioned in our NCERT book are all blunt end, they are all staggered end cutting, okay, or protruding tail cutting one. All enzymes, okay. The first enzyme that Werner Arber and his team isolated, hint 2, is a blunt end cutting. That is why it is not mentioned in our book. So all others are staggered end cutting. Hint 3 onwards. Okay. We are having XXO. What is that XXO? Xanthomonas horsicola. Xanthomonas horsicola. Its sequence is CTC-GAC. It cut between C and T. So it is cutting here and the other one here. So it is a staggered end cutting. Haemophilus influenza decom. That is hint 3. Hint 2, blunt end cutting. Hint 3, staggered end cutting. That is the cutting sequence, recognition sequence. And style 1, Streptomyces albus. Streptomyces albus, GTC, GAC, cutting between G. So all of them are cutting like that. Whereas E. coli, that has been asked twice. Okay. Last year and before, I think it is 2017 also this was asked. Escherichia coli 5, E. co R5, not E5, R5, GAT, ATC. That is the sequence, and it is cutting between T and A. So it is in the middle of the palindromic sequence that they are cutting, blunt end cutting. So keep it in mind. Echo R1 is given in your book, Echo R5 is not given in your book. Okay. So it is cutting in the middle of the palindromic sequence, blunt end cutting, Escherichia coli 5, Echo R5. So this is the answer. Again, DNA fragments. See, we are cutting the DNA with a restriction enzyme. It will be cutting wherever there is a recognition site, cutting site for that enzyme. It will not be cutting as and where we want or we wish. It will be cutting only where there is a cutting site. And these pieces are separated based upon their sizes. Okay. Because small sized molecules will be traveling faster in an agarose gel because of the crystal lattice nature. Through the gaps, small gaps in between the crystal lattice, smaller molecules will be squeezing. That is why smaller molecules will be moving faster than larger molecules. See, movement is always towards anode, positive charge. Why they are all moving to positive charge? All nucleic acids are negatively charged. All pieces are negative. So it is not based upon the charge. They are moving to the anode because they are having negative charge. That's why they are attracted towards the positive charge. And DNA fragments are all positive charged? No. They are all negatively charged. Correct. They are neutral, either positive or negative. No. Then why they are all moving at different speed? Smaller the molecule, greater will be the speed. See, suppose there is a crowd. A large sized vehicle cannot move through that crowd very easily, but a small sized molecule or small sized vehicle or a small sized man can squeeze between the crowded space very easily, more comfortably. Same principle. Smaller the molecule, greater will be the speed because they can move faster, quicker. The process of separation and purification of expressed protein before marketing. So the overall process whereby the expressed protein, that's called recombinant protein. That recombinant protein is purified, is marketed, is called downstreaming processes. It is called downstreaming. Bioprocessing is processing of living things, their molecules, etc. It is not post-production, but it is downstreaming. Okay, from a downstreaming port, we are all using or we are all taking out used cultural growth, and from that cultural growth, we are all separating and marketing. The entire process called the downstreaming process. Okay, it's a very important event because the downstreaming process should be very carefully done, otherwise there will be a lot of waste. It may be sometimes very expensive and we may be incurring a huge loss of the finished products also. DNA fragments separated on an agarose gel can be visualized after staining with. What is the stain? We are using ethidium bromide. This ethidium bromide will be attaching with the double-stranded DNA only, not with a single-stranded. Okay, so DNA is a double-stranded one. It gets attached to the double-stranded DNA only. That's why we are using ethidium bromide. Since ethidium bromide is a fluorescent dye, it can be visualized only under UV light. Okay. Now, what is the criterion for DNA fragments? Movement on agarose gel during gel electrophoresis. Remember, the same point is asked repeatedly in several years. These are all NEET examinations questions asked in previous years. Okay. So, what is the criterion? All DNA fragments are having the same charge, negative charge. Then what is the criterion? Their molecular size. Larger the molecule, either in size or in molecular weight. Smaller the fragment size, farther it moves. It moves farther and farther away from cathode. See, we are loading it in the cathode. We are loading here. It is moving farther and farther away from it. If it is smaller in size, okay, smaller the fragment size, farther it moves, great distance it covers. Larger the fragment size, the farther it moves. No, positive charged fragments move to the farther end. No, all of them are negatively charged, which is not positive. Negative charged fragments do not move. No, it is not like that. All nucleic acids are, and their fragments are negatively charged. There are acids, there are negative charge. That's why they are all moving towards the anode. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? See, we are all using different types of vectors. As I told you, we used in earlier times only simian viruses, SV40, for introducing foreign DNA to human cells. See, we cannot use plasmids because plasmids will enter into the bacteria cells. Here, the host cell to which we want to give a donor DNA is human lymphocytes, blood cell. For transferring to human, we will be using retroviruses. Nowadays, we are using disarmed pathogens. We know that retroviruses are highly dangerous, but this enemy can be made our friend, our foe can be made a friend. How? By removing that harmful part. Okay, a retrovirus, we have studied it in detail. A retrovirus will be having a size sequence and a packaging sequence which contain 3 coding sequence for viral qualities, Paul, gag, N, etc. This is the packaging sequence, and this is the what is called the viral genes. Which is responsible for the viral properties, viral genes. Okay, we just remove these viral genes. Okay, and thus we can convert this one as our friend, and we have used this one. Okay, in treating, in the next chapter we'll be studying Ashanti De Silva, the first girl to be treated for severe combined immunodeficiency disease. Okay, this severe combined immunodeficiency disease was treated by transferring normal genes. That's called gene surgery. Okay, we are all surgically removing defective genes and replacing with healthy normal genes. Gene surgery. Okay, for that we use retroviruses. In early stages, we used alpha or lambda phages. They were all used for transferring DNA for animal cells. TA plasmid from Agrobacterium tumefaciens, it is causing tumor, but only to dicot plants they will be getting infected, broadleaf plants. So, dicot plants. It is also a pathogen because it causes crown gall disease, a cancer. So, we are taking it. This TA plasmid is having a particular type of DNA called tDNA. Now, we take it out. Okay, remove the tDNA and replace with our desired DNA. Okay, this tDNA is cut and removed, and in its place you add our desired DNA and give it to a plant cell. It will faithfully transfer that DNA, desired DNA only into the genome of the host. That's why this type of tDNA containing TA plasmids of Agrobacterium tumefaciens is called nature's genetic engineer. It can give that DNA that is given to or artificially given to this tDNA position. This is the desirable DNA that alone will be given to the genome of the host cell. Both of them are dangerous. Retroviruses and Agrobacterium tumefaciens are dangerous pathogens, but we have a technique now to make them our friend. They are all disarmed now, and such disarmed pathogens are now our friends. So, this technique is called disarmed pathogen technique. Clear? PBR is an artificial Bolivar and Rodriguez synthesized and is used for transferring DNA to bacteria. Okay? So, here the question is to human lymphocytes. Ashanti De Silva was treated by using 2 varieties of retroviruses. one helper and the other one the vector retroviruses. Okay. Again, we are having the same question in another form regarding PCR. The correct order of steps in polymer chain reaction. See, it is because of the PCR now we are having a chance of having our human genome project done and having gene banks produced. Okay. Kyrie Mullis, his machine, thermocycler, as I told you, is having 3 steps. These are the 3 steps. In step A, the temperature is increased to 94 degrees. The DNA, original DNA that we want to have amplified, DNA amplification is done. It is separated into 2 strands. The 2 strands are separated. That separation is called denaturing. Okay. So the 2 strands are denatured when the temperature is increased to 94 degrees. Now the temperature is brought down to 50 to 60 degrees. When the primers, what are primers? Small molecules of single-stranded DNA, which we have artificially produced 2 copies. They become attached to the 3' end. Always primers will be attached to the 3' end. Okay. That is called primer annealing attachment, so that it becomes double-stranded for that part. Now, temperature is increased to the optimal temperature needed for the working of tag polymerase enzyme, 72 degrees, so that one by one the nucleotides are all attached and chain is elongated. And now it become, each of these single chain become double chain. Thus, from one molecule, now you are having 2 molecules. They are multiplied, okay, in a logarithmic way. They are all multiplied. So 1 become 2, 2 become 4, 4 become 8, like that. In every cycle, the number become multiplied. Okay, so what are the 3 steps? We are having denaturing, primer annealing, and chain elongation, which is called extension. The chain is extending, growing. That's called chain extension. How the chain extension is occurring? Because of the action of tag polymerase. See, tag polymerase is acting only in the last stage, 3rd stage, so that single chain become double chains. So it's a cyclical event. That's why I have drawn like that. It's a cyclical event having 3 steps. Clear? DNA precipitation out of a mixture of biomolecules. How you will be taking out the DNA from a source? It may be a plant, animal, or a fungus, or a bacteria. So we have to treat them according to the nature of the cell wall. If they are having a cell wall, okay, remove the cell wall, then centrifuge them under ultracentrifugation, so that the cell membranes will be bursting and the content will be coming. And one by one, by usual treatment, by you using proper chemicals, you remove the heavy molecules like protein, carbohydrate, lipids, RNAs, etc. And what will be remaining will be only the DNA. Because all others have been separated by using special techniques, by centrifugation. And finally, what is remain will be only the DNA. The DNA is a long polymer, heavy molecule, and add chilled ethanol. Adding of chilled ethanol will make them to develop into long fiber-like structures, thread-like structures, and using a copper hook-like structure, you can separate it out. This is what is called spooling. The technique is called spooling. And we add chilled ethanol to precipitate DNA out of that mixture. only DNA will be precipitated, and so we can isolate the DNA from that mixture. Okay. And coming to the last question of the day, following steps describe the characteristic of the enzyme restricted endonuclease. Identify the incorrect statement. Remember, a restricted endonuclease will recognize a particular sequence, which is called its recognition sequence, and will cut within that recognition sequence. Actually, there are 3 types of restriction enzymes based upon how they will be cutting. Type 1 and 3 will cut outside their recognition sequence. That is why we don't want it. We don't use it because they are all cutting away from the recognition sequence. Whereas type 2 alone will cut within them. That is why we use it. Okay, we use type 2 recognition enzymes, which are all recognizing a sequence and cutting within that recognition sequence. Okay, and how they cut, whether they are cutting within the middle, that is called blunt end cutting, or they are cutting away from a middle, that is called blunt end cutting. Staggered end cutting. So, we use type 2, and among type 2, we are having blunt end cutting and staggered end cutting enzymes. Okay, I have given you a example where they are all cutting. Staggered end cutting, EcoR5, blunt end cutting. Okay. Now, the enzyme binds DNA at a specific site, correct? That is called recognition site, and cut only one of the 2 strands. No, they will be cutting both strands. Both the strands are cut. This strand and this strand are cut. Both the strands are cutting. If one strand is cutting, they are called nicking enzymes. Only one end. Okay, one strand is broken. The enzyme cut the sugar phosphate backbone at specific sites on each strand. No. See, the enzyme cut sugar phosphate backbone. What is sugar phosphate bone? The nucleotides, adjacent nucleotides are connected together by ester phosphate bond. That is the correct one. So, enzyme cut sugar phosphate backbone at the specific site of each strand. The enzyme recognizes a specific palindromic nucleotide sequence on the DNA. They are recognizing a specific sequence. That specific sequence is a palindromic sequence. And it is within that palindromic sequence, they are all cutting. I told you it is type 2. Palindromic sequence, correct? The enzyme cut DNA molecule at identified position within the DNA. They are cutting at identified. They are not cutting anywhere. They are recognizing a specific site. That is called a palindromic site, which is specific for a particular enzyme, which will be usually having 4 or 6 nitrogen base sites. And within that recognition site, they are all cutting in a specific fashion. Even there is a specificity where they are all cutting between first and second one, second and third one, like that. There is specificity. Clear? So that also is. So the first one is the wrong one. Clear? Thus, we have completed the discussion of important questions from the chapter Biotechnology 1. Understand, we have to discuss each and every word in our NCERT book. You should be thorough with every word, not even every sentence. Every word should be thorough to you from this chapter, as is the case of any other chapter. So, you have to read it, understand it correctly, every word, don't omit even a single sentence, and make it thorough, understand it thoroughly. Okay? And with these words, I conclude my session, and I am stopping the discussion of Biotechnology 1, and in next day, I will come with the Biotechnology 2 applications. Okay? Until then, goodbye. Wish you all good luck till then. See you. Thank you all. Thank you very much.